With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
So the question is radioactive element used to label DNA bacteriophage in Wareing-blender experiment of Hershey and Chase was S35, P32, N15 and C14. So we all know that Hershey and Chase were a series of experiments conducted in 1952 by Alfred Hershey and Martha Chase that helped to confirm DNA is a genetic material. Okay? While DNA had been known to biologists since 1869, many scientists still assumed at the time that proteins carried the information for inheritance because DNA appeared to be an inert molecule, and since it is located in the nucleus. Okay? Its role was considered to be phosphorus storage. In their experiments, Hershey and Chase showed that when bacteriophages which are composed of DNA and protein infect bacteria, their DNA enters the host bacterial cell, but most of their proteins does not. Hershey and Chase and subsequent discoveries all serve to prove that DNA is the hereditary material. Since phosphorus is contained in DNA, okay? Phosphorus contained in DNA, but not amino acid. Radioactive phosphorus P32 was used to label the DNA contained in T2 phase. So here option B is correct. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.